When modern medicine attempts to enhance cognition, the default mechanism is saturation. Traditional interventions typically flood the brain with metabolic precursors or force electrical stimulation to push the central nervous system into overdrive. Phosphatidylserine, or PS, operates on an entirely different biological mechanism. It is an endogenous, negatively charged phospholipid, a fat molecule produced naturally within the body. Phosphatidylserine targets the foundational architecture of itself, prioritizing structural integrity over metabolic speed. Looking at this structural formula, we can isolate exactly where this molecule does its work. PS resides almost exclusively in a highly specific region, the inner leaflet of the neuronal cell membrane. Any clinical improvements in memory consolidation or recall are direct downstream effects of its physical presence in this specific membrane layer. To understand how this single molecule dictates brain power, we will map its membrane interactions, track its volatile history of isolation, and define precise clinical dosing parameters required for human efficacy. Optimizing high-level cognitive processing requires clinicians to first optimize the microscopic structural bedrock of the cell. We have to zoom in to the microscopic level to examine the default architecture of a single neuron's lipid bilayer. This 3D cross-section illustrates membrane fluidity. A rigid lipid bilayer accelerates cognitive decline. By inserting itself, PS acts as a physiological lubricant. Its specific shape prevents surrounding lipid tails from packing tightly, maintaining flexibility so crucial ion channels and receptors shift without structural resistance. This lack of physical resistance connects directly to the biological logic of memory formation. A high concentration of PS physically facilitates vesicular release. Vesicles carrying key neurotransmitters, acetylcholine and dopamine, merge easily with the cell wall, emptying into the synapse. Alongside neurotransmitter release, PS provides the necessary structural scaffolding for nerve growth factor receptors, enabling them to anchor securely and function properly. A patient's memory retention capacity is directly throttled by the physical flexibility of their neuronal membranes, a metric where phosphatidylserine serves as a critical, localized regulator. The mechanics of active thought only represent half of the molecule's utility. We also have to look at the life cycle of the neuron, specifically the process of programmed cell death, known as apoptosis. Clearing dying cells is an absolute biological necessity. Failing to remove them leads to severe localized inflammation and disrupts the surrounding neural network. In this cross-section of a compromised cell, the trigger for clearance is the physical translocation, or flipping, of PS molecules from the inner to the outer layer. Once exposed on the cell's exterior, PS acts as a highly specific chemical signal, flagging it for immune removal. This precise receptor binding safely initiates phagocytosis, prompting immune cells to consume and clear the compromised neuron before it degrades. As the brain ages and PS concentrations drop, this signaling mechanism is impaired, leading directly to an accumulation of toxic cellular debris. PS serves dual biological roles. It facilitates the immediate actions of active cognition and serves as the precise chemical trigger required for long-term neurological waste management. Given this vital structural importance, the medical community had to figure out how to isolate the molecule for human supplementation. In the early 1990s, the first clinical trials established profound cognitive efficacies by extracting PS directly from bovine cortex tissue. That progress halted. The industry abruptly pivoted away from bovine sources due to the emerging threat of transmissible prion diseases, specifically mad cow disease. Medical regulators scrambled to find an alternative extraction source that maintained cognitive efficacy without the risk of spongiform encephalopathies. The modern pharmacological solution was to extract bioequivalent phosphatidylserine entirely from plant sources. This side-by-side molecular diagram highlights the structural differences between the two sources. The variation lies entirely within the composition of their lipid tails, attached to the core glycerol backbone. This brings us to the concept of biochemical bioequivalence. These variations in the fatty acid chains do not alter the core function of the molecule head. Despite differing origins, modern plant-derived PS interacts with the human neuronal membrane in exactly the same way as the historical bovine extractions. Surviving the prion crisis confirmed the clinical value of the molecule, forcing science to engineer a safer, infinitely scalable plant-derived alternative. To accurately define its utility, we need to compare PS directly against the wider landscape of lipid-based nutrients. Unlike enzymatic precursors such as CDP-choline, PS provides pure physical scaffolding. It does not chemically stimulate the brain. It builds the environment where stimulation happens. This also clears up the frequent clinical confusion between PS and omega-3 fatty acids like DHA. This split-screen diagram highlights their distinct roles. Omega-3s act as the generic, raw building blocks for the overall membrane. PS, however, provides the highly specific architectural organization needed to cluster and build active receptor zones. Neurooptimization relies on synergistic dependence. The raw materials provided by omega-3s cannot effectively support cognition without the architectural framework of PS to organize them into functional pathways. Translating this biological data into a daily routine reveals how advanced medical practitioners prescribe PS to address age-related decline. This clinical dashboard outlines the standard target. The daily dosage required to achieve measurable cognitive support falls strictly between 100 milligrams and 300 milligrams. Timing protocols are dictated by the molecule's half-life and its secondary, clinically verified ability to blunt cortisol production. Clinicians generally advise administration during the evening or immediately post-exercise to maximize this exact cortisol modulation effect and support recovery. Sourcing logic comes down to the individual patient. Choosing between sunflower and soy lecithin derivatives is based entirely on avoiding specific individual allergen sensitivities. These protocols are designed as long-term habits. The fleeting effects of acute chemical stimulants spike and crash, whereas PS requires a slow, permanent structural intervention. The clinical consensus aligns with the cell biology. Proper PS integration reliably supports memory consolidation, processing speed, and baseline neuroprotection over the lifespan. Reversing cognitive decline and optimizing processing power does not begin with artificial electrical stimulation. It begins by securing the fundamental structural bedrock of the cell membrane.